Let's come to the next talk by Doreen Becker, and she will give an introduction in the structural and functional characterization of 5 bovine cell lines. Ist auch so ein Ding da liegen, oder?
Ach, da. Sorry. Yeah. So the microphone is on. Hi. So thank you very much for the introduction. And in the next couple of minutes, I would like to talk to you or present to you some of the data that we generated in regards to the structural and functional characterization of 5 bovine cell lines. But before I do that, I really want to take the opportunity to thank all the partners that contributed to this work package. It took a lot of effort to get all the cell lines ready and all the data generated. So thank you. But now let's come back to the cell lines and let's see why we are actually interested in the characterization of cell lines. So, well, cell lines are a very interesting model to study the genetic features and the regulatory genome of the species of interest, and therefore, and also for example, for ethical reasons, they are used as surrogates for in vivo experiments. And I'm sure we are all using cell lines that we think we know a lot about them. So we know some phenotypes and we know maybe some reactions to certain stressors that those cell lines present, but usually for the functional annotation, we don't know anything. And it was shown that cell lines so when we look at the functional annotation, it was shown in recent years that the functional annotation is influenced by the ploidy of the cells, and it was also shown that cell lines that became immortalized showed a high amount of aneuploidy and also heteroploidy. So therefore, it is really important to molecularly characterize the cell lines so that we know a little bit more about them. And that was actually also the main aim of this work that I will present here. So we had access to 5 bovine cell lines. We had the EBL cell lines that was derived of a bovine embryonic lung. We had trophoblast cells that were trophoblast cells named F3 that were derived from cotyledon, and we had the MDBK cell line that were established from a kidney of an adult animal. Those 3 cell lines spontaneously immortalized, whereas the MACT cell line was derived from memory cells that was immortalized using an SV40 antigen. And then we also had access to the luteal cells that were established by Abner, and you already heard about the characterization of those cells. So in order to achieve the goal of gaining insights into the functional annotation, we used the whole genome wide omics approach, and in this talk, I will only focus on the whole genome data, the expression data of the cell lines, and a little bit on the ATAC-seq data, so the chromatin accessibility of the cell lines. I wanted to say that those cell lines were cultured in the same lab by the same person, and the DNA or the RNA or whatever we needed was isolated and was distributed to the partners in order to generate the data. So when we look at the genomic, at the whole genome sequencing data, identifying SNPs and small indels, we could see that the cell lines are quite similar in their numbers, with the luteal cells having the highest amount of small indels and the F3 having the highest amount of SNPs. On average, we identified 7.8 million SNPs and 1.45 million small indels, but you couldn't really see a difference between the cell lines in general, and also you couldn't see any difference between the transfected and between the one transfected and the one the spontaneously immortalized cell line. We then also wanted to look at the genetic structure of those cell lines, and therefore we used 2 criteria. So one was the read depth. So if we, if chromosomes would show the same ploidy, you would expect that the coverage of each chromosome is kind of similar to the average chromosome of the whole genome. And if the coverage is much lower or much higher than the average coverage, that is a sign that there might be a change in ploidy. In addition, we use the ratio of the wild type and the alternative alleles for heterozygous SNPs, and this should be around 0.5 for diploid chromosomes. So we did that with all cell line, but because of the sake of time, I'm only showing you one example for the EBL cell line. So what you see here is a circ plot and a visualization of the of those 2 criteria. So you see the coverage plotted for each chromosome in light green and the allele ratio in light orange. Now, if you zoom into a chromosome, what you can see here is that at the beginning, the coverage of chromosome 1 is actually quite similar to the average coverage of 50x. And also the allele ratio is on average of 0.5. But then at the end of the chromosome, you see an increase of the coverage for the rest of the chromosome, and you also see that the SNP ratio varies between 0.25 and 0.75, which indicates that this chromosome is partly triploid. If we look at, for example, for chromosome 11, there you see that the coverage is actually increased throughout the whole chromosome, and the allele ratio varies throughout the whole chromosome, indicating that the whole chromosome is actually probably triploid. Then we did that with, again, as I said, we did that with all cell lines and looked at the average coverage. So on these plots, you see the red line that indicates the average coverage for each cell line. And you can see that there are a couple of regions or even whole chromosomes that are either lower or higher than the average coverage. At the end, we had kind of genetic maps, like you see here as an example, where we could indicate, okay, where are the regions that are either triploid or haploid, or have a change in the ploidy at all. So in summary, we saw that in all 4 cell lines, or at least in the 4 cell lines that were already established, we find aneuploidic chromosomes and also a large amount of structural variations. And even for the luteal cells that were again in passage 3, we see that we could already find a high number of structural variations there. When we look at the expression activity of the cell lines, we found that between 13,200 and 15,500 genes were expressed per cell line, but there was also not anything that stood out. But we were interested in investigating if the expression activity is actually influenced by the ploidy of the cells. And so we plotted the median TPM for some diploid chromosomes and for the aneuploidic chromosomes in the example of the F3 cell line. And what you can see here is that the diploid chromosomes 1 and 2 show a similar TPM, median TPM in comparison to the average median TPM for all chromosomes. But what you also can clearly see is that the haploid chromosomes have a decreased expression level, and the triploid chromosomes have a higher expression level. And this is actually true for all cell lines, so we could find that for all cell lines. We then were looking at those chromosomes that had structural variations, partly structural variations, and plotted the region that had a ploidy change in comparison to the diploid region. And what you can see here as well is that the regions that were haploid actually showed a less median TPM, and they showed a less expression. in the region, but that is not so clear for the triploid chromosomes or for the triploid chromosomal regions. So you can see that chromosome 1 and 14, for example, they have an increased expression level, but chromosome 5 is slightly reduced, and even more pronounced is the region on chromosome 18, which is where the expression is reduced. As you heard from Gabriel already, we had a lot of tissues that we sequenced, and here is a heat map that Gabriel provided. And the only interesting thing is the things that I highlighted. So you can see the heat map of the transcriptomes of 3 of the tissues that are the tissue of origin for 3 cell lines, so lung, kidney, and ovary. And as you can see, those have a similar expression pattern, and so they are clustering together. Now the cell lines, they form a cluster on their own, and they actually don't show any correlation with their tissue of origin. Yeah. Now coming to the epigenomic data for the chromatin accessibility. So we found around 86,000 to 127,000 peaks per cell line, and we kind of were interested if the ratio of the number of peaks by the number of genes is actually also reflected in those aneuploidic chromosomes. So we again plotted those ratios in diploid chromosomes and in the chromosomes with other ploidy. And here you can see that, like the expression pattern, that haploid chromosomes show a reduced ratio, and triploid chromosomes show an increased ratio. However, it is not as clearly as with the transcriptomic data. We also did that with the chromosomes with structural variation and plotted those. And here the picture is less clear, I would say. So you have chromosomes where you actually can, or the chromosomal regions where you can see that the ploidy affects this ratio, but you also have regions where you can't really see that if you just look at the picture. So the chromosomal structural variation is only partly reflected in the chromatin accessibility. In summary, we could show that the structural variations, large structural variations, are very common in cell lines, in all those cell lines that we characterized. We use different omics data, and as Gabriel said, we have a lot of more data, and hope that we can even further detail the genome functional annotation of those cell lines. And you can say that the chromosomal variation is reflected in the transcriptome and also partly in the chromatin accessibility. So thank you for your attention, and I'm happy to take questions.
Thank you, Doreen. We even have some minutes for discussion. Also, online participants, raise your hands, please. Krishna. Microphone.
Krista Kühn, thank you, Doreen, for this talk. In addition to what Abner just said about the number of passages, do you recommend that in the future the different laboratories working with the cell lines also report the aneuploidy level? So before starting with the cell line experiment, do some resequencing of the cell line before you report anything?
I don't know if you really need to do the resequencing if you would use the same cell lines that we characterized now, but I would do that for all cell lines that you are using at the moment, especially when you use them very frequently. And it's not only the reporting of the ploidy, but also of the expression level in the comparison to the tissue of origin and all that what Abner already suggested. So the passage number takes a huge effect. We already saw that passage 3 for the luteal cells had a huge effect on the genetic structure. I mean, it was passage 3. and it already showed 7 structural variations, which I think is quite impressive. So yeah, you should report everything if possible.
If I may add, given the fact that these cell lines are frequently distributed between laboratories, so can we establish kind of inheritance lines? And maybe if you assume, for example, you receive a cell line which has been passaged for, let's say, 10 passages in lab A, wouldn't it be wise to do some resequencing then yourself?
Yes, it would totally, because we already saw that there is also a difference in not only in the passages but also in the different protocols. So depending on the protocol you are using for the culturing, you will have different results, especially affecting the expression level. Maybe not so much the genetic structure. So it is important, to know also note where the cells are coming from. So where you received your cells, maybe. It was already shown in other cell lines that you have several subgroups of different populations of the same cell line. For example, for the Hep G, which is a cell line.
So maybe in the future for open data, then journalists should ask not just for the results, but also for the sequence of the cell line.
Yeah, and we should get funded to do that.
Yeah, this is one problem. The other problem is that some people work with cell lines which are not the cell lines they think to work with.
Yeah, that's just
another issue.
Just to comment on this, we have one example in Bovrec where we, from a rather renowned laboratory, received a sample from bovine, which turned out to be from dogs.
Yeah.
So, I think we are well in time. Thank you very much, Doreen.